Hello and welcome back to the Biochemistry for Health Sciences channel. Today's video is on the pentose phosphate pathway. In previous videos we talked about main street. Just to remind you, that's how we take glucose, send it through glycolysis, and then the PDH reaction, and then the TCA cycle, eventually going through the electron transport chain and oxidative phosphorylation to give us ATP or energy. Since glucose is the most important fuel in the human body, this main street is very critical, has to happen 24/7 in order for our cells to generate energy. So the pentose phosphate pathway that we'll call PPP is right down here. We are taking away glucose 6-phosphate from main street and sending it into the PPP, which is also called HMS. In recent videos, we saw how we take citrate. You can see that's one of the intermediates of main street. Send the citrate out of the mitochondria into the cytoplasm, and then break the citrate down into acetyl-CoA. And then we take the acetyl-CoA and make it into fatty acids. That's fatty acid synthesis. And then combine the fatty acids with glycerol to give us triglycerides. And the acetyl-CoA can also be made into cholesterol. So in both these processes, you can see NADPH is very important. We need a lot of reducing power to make fatty acids, triglycerides, and cholesterol. And this reducing power comes in the form of NADPH. So in this video, we'll see that HMS or the PPP provides most of this NADPH that is required for synthesis of various biomolecules, especially the lipids. So if you look at the structure of the coenzyme NAD and compare it with NADP, you see they're more or less the same, except there is a phosphate here. You can see there is a phosphate attached to the sugar in NADP. So although the structure is very similar, the functions of these coenzymes are very different. NAD is used for oxidation of various metabolites. NADPH, the reduced form, NADPH, is used as reducing power for biosynthesis. And then we'll see in a few minutes that NADPH is also very important for scavenging as well as the generation of reactive oxygen species. So let's look at the key steps of this pathway that is called the pentose phosphate pathway, or PPP. Now we see glycolysis going in this direction, and in the first reaction of the PPP, glucose 6-phosphate, which is an intermediate of glycolysis, is converted into 6-phosphogluconolactone. So basically it's oxidized, this carbon here is oxidized to form a lactone, and this reaction is catalyzed by the enzyme glucose 6-phosphate dehydrogenase. The oxidizing agent here is NADP, and what we produce is NADPH. Since we are removing glucose 6-phosphate from glycolysis from main street, we are taking it away. We are basically shunting the glucose 6-phosphate, and glucose 6-phosphate is a hexose, 6 carbons, monophosphate. That's why this pathway is called hexose monophosphate shunt. So we are shunting away the hexose monophosphate, which is glucose 6-phosphate, away from mainstream. This enzyme, glucose 6-phosphate dehydrogenase, is the key regulatory enzyme for the pentose phosphate pathway. Based on the quantity of enzyme found in various cells, and this is done by microarray mRNA expression, the highest amounts of enzyme of this G6-PDS, glucose 6-phosphate dehydrogenase enzyme, is found in red blood cells, in adipocytes, in cardiac cells, smooth muscle cells, as well as in the lungs. So apparently this pathway is very important for these cells listed here. In the next step, the lactone, remember lactone is a cyclic ester. So here is a carbon, this is your COOC, so that's an ester, and this is a cyclic form of an ester. This ester is hydrolyzed by the enzyme lactonase, which is a hydrolase. So when you hydrolyze this ester, this carbon will come out in the form of a carboxylic acid, COOH, and this carbon here will have the OH group, and that's this carbon that will have the OH. So basically, hydrolysis of the ester will give you back your carboxyl and your hydroxyl group. The next step is an oxidative decarboxylation. This carboxyl group comes out as carbon dioxide, and this carbon here gets oxidized from an alcohol to a ketone, and that gives us a 5-carbon intermediate called ribose 5-phosphate. Once again, we see the formation of this very important reducing power, NADPH. So these reactions, conversion of glucose 6-phosphate all the way down to ribose 5-phosphate, this part of the PPP is called the oxidative phase, simply because there is oxidation reactions that are happening. And in the oxidative phase is where we produce NADPH. And notice that the oxidative phase, that these reactions are all in one direction, the arrows are going in one direction. That means this part of the pentose phosphate pathway, this part is irreversible. The oxidative phase is irreversible. In the next reaction, the ribose 5-phosphate is converted into its epimer called xylulose 5-phosphate, and epimer basically means that this hydroxyl group has switched with the hydrogens. So basically that's an epimer of ribose 5-phosphate called xylulose 5-phosphate. Another ribose 5-phosphate is then converted into the isomer. So this is a ketose, is converted into the aldose isomer. Okay, so basically a ketose is converted to an aldose. And this is called ribose 5-phosphate. So this conversion of ribose 5-phosphate into xylulose 5-phosphate and ribose 5-phosphate, this begins the non-oxidative phase of the pentose phosphate pathway. Notice that this reaction is reversible, okay, which means you can take xylulose 5-phosphate and make it ribose 5-phosphate, or take ribose 5-phosphate and make it into ribose 5-phosphate. So here we are making ribose 5-phosphate. Now ribose 5-phosphate is a pentose. There's 5 carbons here. Ribose has 5 carbons. So that's a pentose. Ribose 5-phosphate has a phosphate, so this is a pentose phosphate. So because we are making a pentose phosphate, which is ribose 5-phosphate, we call this pathway pentose phosphate pathway, or the PPP. So PPP or HMS are the same names for the same pathway. The next reaction of the non-oxidative phase of PPP is catalyzed by an enzyme called transketolase. This is a transferase enzyme. It requires thiamine pyrophosphate. Remember, thiamine is vitamin B1. Now, transketolase is an enzyme that transfers a 2-carbon unit from the ketose, and the ketose here is xylulose. So basically, you're taking away 2 carbons from here, and we are transferring it to the aldose, which is ribose 5-phosphate. The ribose 5-phosphate will become from 5 plus 2, they'll become a 7-carbon sugar, while here we have 5 minus 2, this will become a 3-carbon sugar, glycerol 3-phosphate. Once again, this transketolase reaction is reversible. In the next reaction, another enzyme called transaldolase, which is also a transferase, will transfer a 3-carbon unit from the ketose to the aldose. So basically, the cytohepulose is a ketose. We minus 3 carbons here, and we send the 3 carbons to the aldose here. So this will become 7 minus 3. So this will be 7 minus 3. This will give you 4 carbons, erythrose. And then this is 3 plus 3 carbons will give you 6 carbons in the form of fructose. And once again, the transaldolase reaction is also a reversible reaction, as in case of transketolase. In the last reaction of this non-oxidative phase, another transketolase enzyme will now transfer, just like before, 2 carbon units from the ketose, and in this case the ketose will be xylulose, and you'll transfer these 2 carbon units from the ketose to the aldose. In this case, the aldose is erythrose. Once again, this is a reversible reaction. The xylulose with 5 carbons minus 2 carbons will give you glycerol 3-phosphate, which has 3 carbons, while the erythrose, which has 4 carbons, will take these 2 carbons here. So 4 plus 2, that will give you 6 carbons, and that will be fructose 6-phosphate. So you can see that this last reaction, catalyzed by a transketolase, links the pentose phosphate pathway with glycolysis. So this pathway is connected to glycolysis. So the PPP produces 2 very important intermediates or substances. One is your ribose 5-phosphate, and the other is NADPH. Ribose 5-phosphate is the sugar that you find in nucleotides. Okay, so in nucleotides, the ribose, the sugar ribose, and from ribose we get deoxyribose. That's the sugar that we find in nucleotides. So ribose 5-phosphate obviously then is very important for the synthesis of nucleotides, and that is a very important synthesis that happens especially in dividing cells. There's a lot of nucleotides that are synthesized for dividing cells, so that the cells that are dividing can copy their chromosomes, their DNA. Now, another important product of the pentose phosphate pathway that is produced by the oxidative phase is NADPH. So most of the NADPH comes from the pentose phosphate pathway. However, there are other reactions that can produce NADPH. One is the enzyme isocitrate dehydrogenase. This enzyme catalyzes the oxidative decarboxylation of isocitrate to alpha-KG, alpha-ketoglutarate. There are different isoenzymes of isocitrate dehydrogenase IDH. There's IDH3 as well as 2 as well as 1. And yes, you can see they are present in either the mitochondria or the cytoplasm as well as the peroxisomes. IDH3 uses NAD as the oxidizing agent, so that doesn't produce NADPH. However, IDH2 uses NADP and produces NADPH, and so does IDH1. It uses NADP and produces NADPH. Another enzyme that produces NADPH, we talked about this in the transfer of citrate from the mitochondria matrix into the cytoplasm. We talked about this malic enzyme in fatty acid synthesis as well as cholesterol synthesis. So malic enzyme also catalyzes an oxidative decarboxylation where malate is converted into pyruvate. Once again, there are various isoenzymes of malic enzyme. For example, ME1 is in the cytoplasm. It uses NADP and gives you NADPH. ME2 is in a mitochondria. It can use either NADP or NAD. Therefore, it can give you NADPH or NADH. And ME3, which is in a mitochondria, once again uses NADP and therefore produces NADPH. So NADPH can come from various sources besides the PPP from this enzyme here as well as the malic enzyme. So why is NADPH important for cells? As we saw, NADPH is used for reductive biosynthesis, for reduction reactions. We saw that, for example, in the synthesis of fatty acids and then in the synthesis of cholesterol, where NADPH is required for reduction reactions. We'll see some of this also later in future videos for metabolism of nucleotides as well as amino acids, the use of NADPH. Now, besides reductive biosynthesis, NADPH is very important for maintaining the antioxidant capacity or the antioxidant defense of cells. Inside our cells, we have chemicals such as glutathione and thioredoxin, the protein thioredoxin. These chemicals are very important antioxidants. They help neutralize free radicals that are continuously being formed in our cells. So, for example, you can see that in order to break down hydrogen peroxide, we require glutathione, GSH, reduced glutathione. But when this glutathione is used up for converting hydrogen peroxide to something safe such as water, and this is catalyzed by the enzyme glutathione peroxidase, the glutathione becomes oxidized to GSHG. So in order to reconvert the oxidized form back into the reduced form so that it can be used to destroy the hydrogen peroxide, which can be very toxic to cells, we need the enzyme glutathione reductase, and this enzyme uses NADPH to reduce the oxidized glutathione into the reduced glutathione. So the NADPH that comes mostly from the pentose phosphate pathway therefore plays a very important role for cells that have to face a lot of free radicals. And this is, for example, what we see in red blood cells that carry a lot of oxygen. Oxygen, of course, can produce a lot of free radicals. And this is also important for liver cells because liver cells detoxify a lot of chemicals that come from your diet. But in general, this reaction is very important for many cells because free radicals are always produced in our cells simply because the cells use oxygen for generating energy. So oxygen is very important because it generates a lot of energy, but unfortunately oxygen can also produce free radicals. Now thioredoxins are small proteins which help convert the oxidized form of the protein into the reduced form of the protein. So this reduced thioredoxin will help convert the oxidized form into the reduced form. In the process, the thioredoxin becomes oxidized and therefore it has to be reconverted back into the reduced form. And that is done by the enzyme thioredoxin reductase, which requires the reducing power in the form of NADPH. And once again, the pentose phosphate pathway provides most of this NADPH. So this is another very important function of NADPH, and that is to maintain our antioxidant defense, basically to neutralize free radicals. Now, very interestingly, NADPH is also connected to free radical generation. For example, in the generation of superoxide. Now, we do see this as an advantage. For example, in white blood cells, when they ingest a virus or bacteria, in order to break down the bacteria, the virus, or whatever the white blood cells ingest, they have an enzyme called NADPH oxidase, or NOx. And this enzyme plays a very important role in phagocytosis. So the white blood cells actually use NADPH with the help of this enzyme NADPH oxidase to generate superoxide. So here we are actually generating superoxide, and then this superoxide will help break down this virus or bacteria together with other kinds of free radicals that are produced in the phagolysosome. Of course, this inflammation these white blood cells are very important because they help protect us against these microorganisms. But in chronic inflammation, many of these free radicals can diffuse out of the cells and start attacking normal cells. There is also a connection of NADPH with the enzyme nitric oxide synthase that is present in endothelial cells. These are the cells that line your blood vessels. So this enzyme eNOS, endothelial nitric oxide synthase. So under normal conditions, this enzyme uses arginine, oxygen, and NADPH, and with the help of a cofactor called tetrahydrobiopterin, or BH4, we get these products, citrulline, NADPH, and nitric oxide, which is a very important intracellular messenger. However, when the BH4 is insufficient or inactivated, then this reaction is uncoupled. And what happens now is the oxygen in the presence of NADPH will produce superoxide, will actually produce this free radical. So with this background, you can see that the pentose phosphate pathway may be related to many human diseases. So for example, because it provides NADPH for fatty acid synthesis and cholesterol synthesis, this may be related to obesity. NADPH required for cholesterol synthesis comes from the pentose phosphate pathway, and we know that cholesterol is related to arteriosclerosis, which in turn is responsible for the various cardiovascular diseases, including myocardial infarction and strokes. And then the NADPH, as we saw, is important for our antioxidant defense, important for our immune function, but may also generate free radicals. So So this whole antioxidant free radicals relationship is very important for many diseases that we'll talk about in future videos. So because of these connections here, we can clearly see that the pentose phosphate pathway may play a very vital role in several human diseases, and there's a lot of research going on in this area. So what would happen if the pentose phosphate pathway did not occur? So for example, there is a disease called glucose 6-phosphate dehydrogenase deficiency, or G6PDH deficiency. In these patients, one of the main characteristic or feature that we see is hemolytic anemia. So the red blood cells get broken down quite easily, and without enough red blood cells, of course, there is paleness, since red blood cells carry oxygen. Without enough oxygen, there's not enough energy generation and fatigue. When red blood cells are broken down, the heme gets converted to bilirubin. Excessive amounts of bilirubin causes jaundice. So since the red blood cells do not have enough NADPH, they cannot regenerate the GSH, and therefore a lot of free radicals will accumulate in these red blood cells and cause the red blood cells to break. So this is the main feature of G6PDH deficiency, but research has shown that these patients have an increased incidence of diabetes. Now, could that be connected to the oxidative stress that is related to PPP? Could that be connected to lipid metabolism that is closely associated with pentose phosphate pathway? We also see increased kidney dysfunction in these patients, as shown by increased creatinine levels in their serum. And this might be related once again to the antioxidant free radical connection. Now, surprisingly, these patients, more recent research has shown that they tend to have a decreased coronary artery disease. Is that related to decreased triglyceride synthesis and cholesterol synthesis because they aren't producing enough NADPH that is required for the synthesis of these lipids? Well, these are questions that need to be answered with more research. So before we go, let's try to answer one more question. In these patients, nucleotide synthesis is not affected. So why is that? So remember, for nucleotide synthesis, we need this chemical here, ribose 5-phosphate. And without this enzyme, glucose 6-phosphate dehydrogenase, this part is not going to happen. The oxidative phase is not going to happen. Okay. However, the non-oxidative phase is reversible. So basically, we can take these intermediates from glycolysis, use your trans-ketolase and your trans-aldase enzyme, and make ribose 5-phosphate, which then can be used for nucleotide synthesis. So that's why, although this enzyme is missing, that only messes up the oxidative phase. The non-oxidative phase is fine, that is reversible, and these patients can still make nucleotides for the dividing cells especially. Okay, so that is some of the more pertinent information on the pentose phosphate pathway, also called the hexose monophosphate shunt, for healthcare professionals as well as healthcare students. Once again, thank you for looking at this video. Until next time, goodbye, and have a happy and a healthy day.